Rappin with ReefBum is sponsored by Polo Reef and Champion Lighting and Supply. Hey, welcome back. My name is Keith, and I'm joined once again by my co-host Dong. Dong, how you doing there today?
Very good, very good.
Hey, it was great seeing you at the Boston Reefers Frags giving event a couple of weekends ago. It's always a fun time, always a good show.
Oh, absolutely, absolutely. I love it.
Yeah, no, there were some good speakers there. Ben Johnson, Chris Meckley were speaking. A lot of great vendors, a lot of great corals, and it's always great to catch up with everybody. And those guys put on a great show. And both you and I are sponsors of the Boston Reefer Society, and we encourage anybody out there to join your local reefing club. If you don't have one, you could join Boston Reefer Society. You don't have to be living in the vicinity of Boston to join that club. So it's well worth it. So Dong, man, I'm really excited about the show tonight. This is going to be a very interesting discussion we've got going on. Our guests are Salem Clemens, who owns Novel Aquatics. We've got Colin Foord. He's a marine biologist, a coral artist, and co-founder of Coral Morphologic. And we have Dr. Braden Tierney. He's the executive director of the Two Frontiers Project. He's a microbiologist and a data scientist, so that's going to be a very interesting perspective from him, and it's going to be a great panel. So we're going to be talking about how coral probiotics can prevent coral disease. But before we bring on our guests, I want to thank our sponsors who are supporting the show. They're very much appreciated. Polo Reef, make sure to check out Polo Reef's latest YouTube videos. The Polo team takes on another crazy month after ICP test results reveal toxic metals. The team continues to solve the issue. The team fixes the mess from the rock slide in the 17,000-gallon tank last month. Oh, boy. All the while tackling lighting issues, saving rare fish, finally fragging giant acro colonies, and exposing the dirty side of reef keeping. Man, that's going to be
I want to see that.
No one at Polo Reef was happy with the smell. Make sure to subscribe to their YouTube channel at Polo Reef for video drop notifications. Champion Lighting and Supply. Many thanks to Todd for hooking me up with this. Dong, you got one of these too. The Reef Delight lens port and coral lens. I'm going to play some video, actually. I know you have some pictures on
oh my goodness,
Boston Reef Society. I took this today, and just before a couple hours ago. Man, oh man, this thing is like pretty freaking awesome. I used to use a top-down view box and stick my camera in that, and it was very cumbersome. This thing, like, I just I put it onto my my phone, the lens. I figured out what particular lens to put on, and it's a beautiful thing. It's awesome. So if you want to get
easy to use.
So if you want more information and you want to pick up one of these things, visit championlighting.com. Thanks again, Todd. Really appreciate it. So
thank you, Todd.
Let's bring on our guests. What's happening there, guys? How are we all doing tonight?
Great. Thanks for having us, Keith.
Thanks for having us on.
It's good.
Yeah, pretty good.
So, like I said, we're really excited about this discussion tonight. I see we have a whole bunch of people that are already tuned in, and I think ACI Aquaculture is in the house there. What's happening there, Mr. Meckley? Corey Page.
Hi, Meckley.
Yeah. All right. We got a whole bunch of familiar reefers in the chat tonight, but so guys, I mentioned the topic tonight is talking about probiotics and how potentially they can be used to prevent coral disease. But let's, for people that are not in the know in terms of what probiotics are, can somebody just give a real simple definition of probiotics to kind of like set the stage for tonight? Who wants to jump on that one?
I think I can help with that one a little bit. And it's funny because there's a long history behind the definition of a probiotic, and it's like incredibly convoluted and ridiculous. But in general, you can think of a probiotic as a living microorganism that in this case is given to an ecosystem like a coral that's meant to do something good for that ecosystem. So it could increase tolerance to disease or heat stress or whatever it is. And when we think of probiotics, we mostly think of bacteria. So single-celled.
Mhm.
Yeah, no, that's good. Just kind of like wanted to lay that foundation for tonight. So, what qualifies a microbe as a probiotic?
Well, it's the fact that it's alive. So it's like biotic referring to life, and so it's a living. It's like you might also hear it as like a live biotherapeutic or something like that, or like a microbial therapeutic. But generally, it's a probiotic refers to like a living thing. So it's as opposed to like, a nutrient you might add to an aquarium or whatever it might be. That would be, it might be considered a prebiotic if it like feeds bacteria. Yep. Or like if you have something that's made by bacteria, that would be considered a postbiotic if you added that in. A probiotic refers to right in the middle. It's like the living thing. So they're really popular in human health as well. If you go to like CVS, you're probably going to see an entire aisle that's just filled with all these things that are usually like yogurt bacteria. People also say that yogurts are natural probiotics, right? And so you can because that's the bacteria there ferment and what actually makes the yogurt. So they're all over the place. It's not just corals, and it's any living little thing.
So we as humans should be consuming probiotics.
Well, that's a tough statement to make. I would say
we don't have to go there. We could exclude the human nature.
The thing that's true, humans aside, right? Like there are very few places on Earth that don't have bacteria in them, right? Like we're covered in them. Like they're filled. Half of your stool is bacteria. Half of the weight, right? Corals are covered in them. They're floating in the ocean. There are like 5 quintillion bacterial cells on Earth at any moment. So arguably they're all doing really important things, right? They maintain ecosystem homeostasis. They eat nasty pollutants and make healthy beneficial things. They do all sorts of really important stuff. They cycle so much carbon in the ocean, right? 20 times the amount of CO2 produced by humans every year. So 20 times that amount is actually consumed by bacteria in the ocean. So arguably everything has probiotics, everything kind of has a relationship with microbes. It just differs depending on the ecosystem, right?
And I think also, it's a good time to point out this concept of the holobiont, right? That, and correct me if I'm wrong, I tell this story a lot in that, we think of there's different types of evolution, right? The type of evolution that we're we understand the most is the Darwinian evolution that takes place between generations, and it's like a sexual reproduction, and you have random chance, and you have natural selection taking place. But in the 80s and the 90s, scientists were studying corals, and they recognized that when corals bleached, and obviously, to go back to the most basic element, corals are the types of reef-building corals that we grow are photosynthetic organisms that have a symbiotic relationship with the zooxanthellae, which is a single cellular organism, an algae, dinoflagellate that's living in the tissue of the corals and is able to provide it with photosynthetic energy, with sunlight or lights in our case, and basically coral bleaching occurs when a coral is stressed out, usually from thermal stress, and heat tends to be the biggest problem that we have. Although cold water can also cause corals to bleach, and so what the scientists recognized was that corals, when they bleached, they could recover, but when they recovered, they might pick up a different type of zooxanthellae in their tissue that in the next summer's bleaching was more resilient to and conferred a level of thermotolerance. And so this was recognized that this is like a whole new type of evolution that was taking place within an organism's lifetime, where the relationship that sort of the macroorganism has with its microorganisms directly corresponds to its health. So, that this was kind of it's kind of a revolutionary realization. And, the way that I describe it is like, once the scientists saw that happening in the coral, we kind of turned the microscope back around on ourselves. And that's when we really realized that, well, just as corals are this complex community of organisms and microorganisms, it's not just the coral animal. Humans are also a whole ecosystem unto itself. And like Braden was saying, like there's, and correct me if I'm wrong, I always hear that there's more non-human DNA than human DNA in our bodies. And like something that I find really fascinating is this, everyone's kind of heard of the metaphor of like trusting your gut or having a gut instinct. It kind of used to just be a metaphor, but like now we know, especially over the last 25 or 30 years, we know that like the microbes in our human gut are actually like producing a lot of the neurotransmitters that get distributed to our brain and ultimately, impact the way that we see and perceive the world. So, what used to be this metaphor is now quite literally a literal metaphor. And this is, now we're sort of living in this world where we know that it's kind of the microbes are really kind of governing so much of of life on Earth. And it's really only in the last couple of decades that we even recognize it. And the work that Braden's doing with True Frontiers is really kind of like looking at these extreme environments where we can find microbes living and trying to figure out if there's a way that it can help human health and now in this case with this project, coral health.
So Braden, do you want to maybe take that as a cue to kind of give us an overview of the Two Frontiers project or Project ReefLink?
Yeah, absolutely. So like the center, the way I like to introduce the Two Frontiers project, and we're a research nonprofit, right? So we go around and we write academic papers and try and understand microbes. But the driving theme is this, the famous, where the line "life finds a way" comes from?
Jurassic Park, baby.
Oh, thank God. Okay, good. Because I gotta say, I've given a few talks at colleges and stuff recently, and like high schools and things, and people say, "Oh, where does that come from?" And it's really
too old. Yeah,
it's so bad. It's really bad. But anyway, life finds a way. At Two Frontiers, we're really interested in how life is finding a way, especially in places where you don't expect it to. So we go all over the world to hot springs at the top of mountains, to hydrothermal vents in the middle of the Mediterranean Sea, to corals that are living next to like acid seeps underwater where they shouldn't really be alive. And we try and say like, okay, how is that happening? Because it kind of shouldn't be happening. And we're really focused on the bacteria that are specifically surviving in those environments, because bacteria, as Colin very eloquently put, they're so important for maintaining ecosystem, what we say homeostasis or health. So they maintain your health, the health of corals. They're also really useful. Like I call them nature's alchemists. They kind of can make anything. They make bioplastics. They can remove metals from the environment. They can do all of this crazy stuff. They can remove all kinds of pollutants. And so we're really interested in going to those highly contaminated places, or places that are really high in methane or high in CO2. And over billions of years, life has found a way to survive off of those things we often want to consume. And in the case of corals specifically, we're really interested in these extreme corals in the oceans, at least. And we'll get into ReefLink in a second. But we've gone to different oceans and places around the world where you have them, living next to CO2 vents, where you have them in really hot waters. Colin has a spectacular example, obviously, of corals living in polluted waters that I'm sure you guys have talked about in the past. We're kind of wondering, okay, how are the microbes playing a role in keeping these things alive? If they are, can that be used to protect corals as they face increasing challenges in the coming years due to warming oceans or polluted oceans? And Project ReefLink is a spinoff of Two Frontiers' Coral Initiative. And this is a wonderful project that we're doing. It's sponsored by Seed Health, which is an organization, a company that's a probiotic company, actually, that's funded some of our work through grants. And of course, with Coral Morphologic, with Colin, and with SitSci, which is a community science-based nonprofit. We're working to basically study aquarium corals because Salem and Colin turned me on to like your entire world and community, and it's amazing because you guys are basically doing these incredible long-term experiments where every single tank is a different experiment with its own microbiome, with their own different corals and own conditions that you're better scientists than like most of the scientists I know. You figured out how to cater these things to keep these incredibly finicky organisms alive. And we just want to know, like, okay, when one of your tank corals gets sick, what's causing it? What are the conditions that let that thing get sick? And can we find a probiotic or prebiotic or any other kind of treatment that can help keep it alive? And so this is a whole community science project. We're basically going to anyone who's willing to send us fragments or sign up and see if we can study the microbiome of these corals. And that's the assemblage of all the bacteria and viruses and other microbial things that are there. And we do that with DNA sequencing and all these in culturing and all these methods I can go into if you really want to hear them. But that's kind of the whole shtick.
So I mean, I know Salem, you've mentioned that it's kind of like a community science project, right? I mean, essentially, reef keeping hobbyists will have the opportunity, if they want to participate, to send in sick corals. Does it have to be a coral that's not doing well?
It can be any kind of coral. And just to be clear, there's like, we've gotten so much enthusiasm for this. It's really amazing. Like we're not funded to sequence or analyze every single coral fragment. So what we're doing is we have a website that I'm sure we could share somewhere where people are able to send pictures of their corals and talk about their tanks and things. And then we're only funded to do a certain amount of analysis. So basically we're trying to build a data set that is some sick, some healthy, controlling for species and conditions, so we can really learn something from this and hopefully get more funding downstream to be able to really scale it.
A quick question. So what kind of a budget are you looking at for this project? In millions or in?
No, not millions for this project. No, this is a lot. You can take a couple of zeros off of that. We're really small. Like, I mean, we're basically, it's a lot of the costs. We are a very small nonprofit. We've been able to keep our costs low by being very creative, much as I imagine as you have to be in the aquarium hobby community in terms of piecing things together for very little cost and figuring out how we can get the most out of the DNA sequencing that we're doing. And so, thinking about it, like, let's say we're going to analyze one coral fragment, right? Okay, so you ship a fragment, we get it. What you then have to do with it is like, we are going to process it. So basically it's sad, but grind it up into a sort of pulp, and you're going to separate it into segments for bacterial culturing. So you need to have all the materials for growing the bacteria that are there. Then also for preservation in our big living database, we call it. So that's cryopreserving what we do, and that's open to scientists for research and all that. So again, we're a nonprofit. And then also for DNA sequencing, right? And so, if we could, it's one of those things where if we could raise millions of dollars, we could analyze everybody's corals and do all of this. But, if you analyze that one fragment, it's on the order, if you take it all the way through sequencing, of a couple $100 a fragment, basically, or if not up to $1,000 potentially, depending on what you're doing and what kind of nutrient analyses. So you can kind of scale if you do 100 fragments, multiply that number by 100 plus the cost of people and shipping and time and all that stuff. And if you can think, it would be wonderful to be able to really, really do this and raise millions of dollars and go for it. And I think that would have a huge impact. And we're hoping to get there. I think we will.
How do you guys differentiate between a coral not being healthy due to, let's say, aquareating flatworms or red bugs, black bugs, or something, versus getting stressed out by something in the system, some instability, let's say, in some key parameters? I mean, there's a lot of different, I guess, areas where corals can get stressed out. How do you kind of put that piece into the
That's a great question. That's a great question. There's one other source of stress I want to throw at you that I'd love to get your take on how we can address this. Salem and I have an experiment we want to do that's like a time series. But basically, if you're shipping us coral, if you're shipping it even overnight, the thing's going to get very stressed out in the little baggie you're shipping it in. That's going to very much change. Like the second you take it out of the water, the microbiome changes. So in like an ideal world, we would, sit in a parking lot, people would bring us corals, and we'd also see what happens if you process a coral immediately, 6 hours after, 12 hours after. And that's, the infinite money scenario where we can have a big sort of process-a-thon. But to answer your question, I kind of look back on you, like what, for me, I'm really interested in bacterially derived disease as a microbiologist, but I'm biased. I would be curious to know like what the biggest problems are, because probably bacteria or prebiotics or postbiotics can help, but I'm seeing some stuff in the chat. Someone's talking about like black crap on the corals or something like that. And like, if that's a huge problem, I would love to sequence the microbiome of corals with that phenotype specifically, because that would be potentially something we could address by changing the environment of the tanks. But I think in terms of actually distinguishing on a very technical note, once you DNA sequence the coral, you're going to get the genomes of any pathogens and the coral and the Symbiotonaceae and the viruses and the bacteria. And that's the kind of DNA sequencing we do. It's extremely high resolution. Think about it as like you're getting every grain of sand on the beach as opposed to just sort of like taking a single handful. And so when we do that, I'll be able to, if there is like an actual eukaryote in there, like you said, a red bug or something like that, I'll be able to say, oh, there's like a big eukaryotic genome that should not be in this sample. And as a data scientist, that's kind of my job, right?
So are these going to be done by your lab, or you're going to outsource it?
So this is all which part of it? The
genome part.
Oh, that's us. That's me. So that's actually my expertise. So my whole training is like all of this. How do you analyze with what we would say machine learning or large statistical modeling or genomic analysis? How do you do that with DNA sequencing data? And if you want this podcast to go on for 12 hours, I can talk about that for the next 12 hours.
Yeah, Chris,
sorry.
Chris Meckley can host that one. So there's a
Salem
question.
already did.
That's right, Salem. You were part of the marathon. You were.
Okay, you guys don't need me here. It's perfect.
Never again.
So here's a question from Sammy Schneider. How will you keep the microbiome from shifting if you're trying to culture? Are you more concerned with identity than respective composition?
So if you're referring to the idea that once you ship a coral or take it out of the tank, the microbiome shifts, I mean, absolutely, you're going to lose stuff. And that's until we come up with a better way, or I can physically go to all of your homes and start culturing immediately from the tank, which I wouldn't put that upon you. Don't worry. And there are going to be those what we would call confounders. However, there is a saving grace, and it's that with culturing, we really pride ourselves on our ability to grow weird bacteria. It is that plus genomic analysis that makes Two Frontiers like really special. I mean, that and the people in a lot of the different projects we have. But from a technical standpoint, it's like those are two things we're really good at, if I may be so bold. And if you have one cell from one bacteria, like just one cell, and you give it the right food, just as like you give coral the right food or give a fish the right food or whatever it might be, you can cause that cell to amplify. It's like natural selection, using natural selection as a tool to grow organisms. So if you ship your coral, if there's some confounding, if something shifts, then like, as long as not everything dies, as long as everything doesn't die, you can theoretically grow out that one thing. But that is really hard. Also, I see a comment about having a mobile lab. We have done that before, and it's so fun. We really, we've done basically extended road trips where we drive around and we do in-field culturing and in-field enrichment and in-field DNA sequencing, which I have a photo somewhere of like running a DNA sequencer on the hood of a Honda Pilot outside in a snowstorm because we were packing up an Airbnb and we were running the sequencer and we had to like keep it going until the last possible minute. And so we're literally getting all of our stuff out of this Airbnb that we were doing all this culturing in the middle of Colorado. And the sequencer is running on the thing. It's hilarious. Anyways, I totally love the mobile lab.
I have a question.
Go
sorry.
ahead,
Go ahead.
Okay. Let's take Rich Ross's question.
Let's see.
And
that's exactly the question I want to ask.
So I guess Rich has a basic question, and Rich is a biologist and very well known in our hobby. So I think, it's just a general question. It sounds like he was on the website, and the question in terms of what exactly do you guys, if somebody does have like a lot of coral, what would you be most interested in getting from a hobbyist that has a lot of coral?
Yeah.
And is there particular types like SPS or LPS, softies? Do you need them all?
So we're mostly focused on, that's a great question. Thank you. And I know that form is very broad. When we wrote it, as you can tell, we went super broad because we weren't sure at all what we were going to get. And honestly, with you guys, Salem has shared this so recently. We've had so many more people sign up that we're still kind of winnowing down exactly what we are going to request. But to that question, like, we're really interested in bacterial disease, generally speaking, on stony corals. Like, I personally have an interest in soft corals, but like for practical purposes, we're really interested in stony corals. And then we have a list of candidate species. I thought if that's not on the site, I can totally update it, but I don't want to rattle it off the top of my head because I'm totally going to get stuff wrong. I don't know, Salem or Colin, if there were other things from those discussions with Krista that you wanted to throw in.
Yeah, I mean, one of the things that I've thought of that at least I've tried to talk to some people about right now is getting elegance corals to look at elegance coral syndrome. There was initial data on that, but I think it'd be interesting to go deeper with this. And it's fairly well described. We know the supply chain with Indonesia, and it'd be interesting to get some fresh ones in and have some of that resistant strain, as Julian Sprung has talked about. Obviously, brown jelly, RTN, STN. And on the flip side, I think healthy versions of all of those corals. So if you're growing some really nice acropora or torches or something, you've not had issues, or you've got some strain you've had in captivity for a really long time, like something like green slimers, for instance, might be decent to look at. All of these resistant types of strains we've farmed for decades now, those are kind of the things, at least in my mind, when it comes to narrowing these down. Then we have the healthy and the kind of resilient strains and what's sick. And then we can maybe figure some things out from those two extremes, potentially.
Yeah. Healthy acropora would be really cool. A lot of our ocean corals are acropora. We also have, again, and forgive me, this is going to very, very blatantly show my naivete, so forgive me. I don't know if you get Porites rus in your tanks at all, or if that's only something, but that is something we're really interested in because we happen to have a bunch of Porites rus from Papua New Guinea that are from right next to acid seeps. And so as acid seeps like CO2, it's 100 degrees Celsius water just bubbling out, and it's just sitting there and it's fine. And then we also have healthy controls. And then there's another study going on in French Polynesia right now off the island of Moorea, where there are a bunch of academics that are pulling Porites rus. And then so it would be cool to get other versions of that or just other Porites to sort of compare how the domesticated microbiome varies. But there's also, everything Salem said is definitely the top priority.
So I have two questions. First question is that because your project is called the ReefLink project, so does Ecotech has problem with that?
Not yet, but thanks for bringing it to their attention, Bob. We really appreciate that.
No, you're totally right. So we went through so many project names, and yeah, we haven't gotten like a letter that says please don't do that, because I think that we actually had some lawyers look into it, and it looked like it was fine. But obviously, if it's a huge problem, this is not a hill that I'm going to go down on. It's perfectly fine.
Yeah,
I don't think they make the ReefLink anymore.
Yeah, they don't have that. They call Mobius now. So
there you go. It's yours.
The next project is a Mobius project.
Yeah,
that's right. Yeah, I'm just following the trend. It's project names is hard to come up with. It was amazing. We went through so many project names because we did a Google of ReefLink. We were like, all right, then someone did that already. Sure, there's something else, and there's, for good reason, so many coral projects out there that it's very hard to find a name that's not trademarked.
So my second question is this project eventually targeting the corals in captive environment, or actually is more toward the coral on the reef in order to save the reef?
Great question. There are so many steps between what we're doing. Let's look at the microbiome and figure out what's there, and we're going to deploy something on a reef in the middle of the ocean. And those steps, as I'm sure you all have thought about so much, are not just like, what's the treatment? It's how do you deploy it? Where do you deploy it? What's the correct dosage? What's all of those things that we don't even know for human probiotics? So this project in my mind is like, I would love to work on closing the loop between sequence the coral, see if there's something wrong with it, identify some candidate treatments, and see if we can figure out how just in an aquarium setting to deploy it, because that would be useful, certainly. But of course, with an eye towards the idea that whatever we do in these smaller ecosystems will be relevant for solving deployment in the ocean downstream as well. But I think, there's a tendency, and certainly in my field of science, to think like, all right, we're going to go straight to the endpoint and solve this huge problem from this very basic question. And I just want to be transparent. Like, I want to figure out the microbiome, get that right. I will, before we do anything else, see if we can help treat disease in a controlled setting. And then, and only then, do I want to think, okay, how can we really scale this? But of course, the eye is towards the ocean. Yeah.
Okay. So I think that to get a government grant, the eye toward the ocean probably is the way to go.
Oh, absolutely, absolutely. I mean, when you write grants, and the introduction,
money
oh,
for
totally.
that.
And when you write the introduction of your grant, you're like, if we do this, so totally.
Exactly.
Yeah.
So because that the final goal is toward the ocean. So what do you think about that the different environment? So if you use the captive environment like a reef tank as a starting point or as a model, so will that be easily applied to the ocean scenario, or is much better to just use the wild coral as the starting point?
So that's a spectacular question. I am of the belief that when you deploy a microbial-based treatment, especially a living one, it should have a clear link, some might say a ReefLink, to the ecosystem that it came from initially. Because like we've seen, I mean, take the human gut, right? I have thought a lot about human probiotics. Like the bacteria that are evolved to live inside us are evolved to live inside us for the most part. Like there's the same thing with mice and non-human primates and all these other ecosystems, and the same thing with corals and their ecosystems. Without having done the microbiology or looked at it yet, my intuition as a microbiologist says, if I find something from your reef coral, I probably shouldn't, and it probably doesn't make sense to actually just go dump it onto a coral in the ocean. What I can learn, however, is the fundamental mechanisms of how bacteria actually interact with the coral, how they are modulating coral health. Like there's a study I really want to do that we might be able to use this data for, that is, and gets super technical fast, so I promise I won't go super deep into it, but you can now fold proteins. You can look at the structures that proteins form, right? And you can see which physical things bacteria make can actually directly bind to the proteins on the host coral cells itself, or the symbiote. And that mechanism of like, there's a microbe that can literally attach to the coral, just like a pathogen attaches to the inside of your gut, or some beneficial bacteria attach to the inside of your gut. That mechanism is going to be conserved, right? So we can learn like, okay, microbe X makes this thing that physically binds a coral receptor. That's going to happen in the ocean and aquarium tank. So we think about an example of that we all probably are familiar with, or maybe is in the human space, it's Ozempic, right? So GLPs, these like drugs that are made for weight loss, actually came from the Gila monster, and they're made from this protein that directly binds human proteins, right? G protein coupled receptors, and that's how they have their effect. So we can totally look at these other environments and say, oh, this microbe in this aquarium coral makes a thing that actually attaches to the coral. That might be relevant in ocean settings in a right controlled deployment.
I think he's really not far off, at least from the existing data we have in the hobby with coral disease and healthy corals using the 16S data from stuff like Aquabiomics. If you compare many of the disease states to what you would see with white band disease, black band disease in the wild, it's the same types of bacterial taxa and the same types of basically ecological niches and transitions that are occurring. So maybe it's not the exact same microbial members, but the kind of movements and transition from healthy to disease, holobiont to pathobiont is a very similar thing. So again, even if it's not one-to-one, there's probably a lot we can learn about the mechanism of coral disease that can be transferred. And then that's the grant money, obviously.
So since you mentioned the white band disease, that actually is something that's very important for the Florida coral. The white band disease has been the basically it is a major focus for like the Smithsonian Institute for the past decade of research for develop probiotic to combat the white band disease. So there's MCH17. That is the one that is they actually even do it, experiment them in the coral reef. The plastic bag method, go there.
Yeah.
So will that be a much faster way to move forward and take advantage of what they have done in the past 10 years? And because that they already identified the probiotic, so why not just improve the MCH17?
Totally. Is that's
whatever
Blake's
already known?
That's Pseudomonas. Blake Eushagima's
That's Blake.
work.
Yeah, that's Blake.
Blake.
Blake's a good dude. Yeah, so that's a great study. Blake, yeah, he's a wonderful scientist and his team's amazing. Yeah, absolutely.
Yeah.
Like there's no reason you can't take forward what's known, while also finding new stuff, because I mean it's like that same argument. I think a lot of the solutions we find are there's no silver bullet, right? There's just no silver bullet for anything. And, so I think that is a spectacular strain to study. Other examples, Raquel Peixoto's work. She's a good friend out in the Red Sea. They have their consortium that's been deployed in the ocean for thermal tolerance. There are a bunch of others out there that are being developed, and absolutely. And we also have stuff we're isolating out of ocean bacteria. And yeah, I just saw Blake a few months ago when we were talking about some of this stuff. So it's really, really exciting. But I think in addition to sort of seeing what's known, I really think it's worth also seeing what else we can find, especially in the pre and postbiotic space, because you're going to want sort of combination therapy, just like you do for human disease.
And to speak on the Pseudo-Altermonas work in particular, all they've proven with those strains is that it can prevent infection. So any active disease, Pseudo-Altermonas inoculation, from what I'm aware of, with those strains, cannot actually treat disease. So once there's already
they can. So yeah, that's the reference right there. I have sorry basically that's a strength of probiotic. They can excrete the antibiotic, so they actually use them to prevention as well as treatment. So, but for this ReefLink project,
that's interesting.
So, for this ReefLink project, I actually see some very interesting financial benefit for the aquarium hobby, because now you're focused on the coral that you collected from the aquarium for the reef tanks. So, you might be able to find something, a probiotic that is marketable, scalable. That's a big market. So for the hobby, because that
I just saw you in the background. Sorry, but sorry. Yeah, go ahead. Sorry.
Because Don, let me just ask one question in terms of what you guys know that's in the marketplace right now. I mean, there are probiotics right now.
Yeah, totally.
Reef tanks. Yeah. What's different from those in terms of possibly developing a new product? I mean,
look, it's entirely possible that we will isolate everything, go through this work, and discover the exact same thing that's in, it could be what Blake found, or like Dr. Thames is one of the big ones out there. That's entirely possible. If I were betting, I would say that's unlikely, just because of the sheer diversity of microbial life in these samples. I mean, more genes in human gut than stars in the universe, right? So there's so more microbial genes. There's so much out there, and there are always strains that do better and do worse. But we're not really in this to we're not trying to sort of we didn't set out to start this project for this project with the goal of we're going to create a competing probiotic or something like that. It was really just kind of let's study the tanks and see what's there. And again, I think it's really worth emphasizing that, you know, we've had a couple of people say, oh, like, I'll gladly pay you to like analyze our samples or something like that. And as a nonprofit, you know, right now, that might change, but right now, we're sharing all the data that we generate, right? Like, it's going to be published. It's going to be put out there. It can be anonymized. That's totally all great. Like, I'm not going to say, you know, this is Keith's tank. Keith has this weird thing in his tank. That's not really what we're going for.
I do have weird things in my tank, though.
Well, that's great. Then I'll go, okay, for you, we'll disclose. It'll be like tank 1, tank 2, Keith's tank 4. But it'll be the control. Don't
we all have weird things in our tanks?
I guess that is the point, I suppose. But yeah, so we're not really set up. So for these samples specifically, we're sharing them. You know, it's all about that. And who knows what we'll find, right? We haven't sequenced yet.
Because that for the probiotics so far available for this hobby, none of them can do what the MCH17 does. None of them has demonstrated has any prevention or cure the coral disease. None. Absolutely zero. Actually, Salem, you have a video about less than a year ago, and you make a very good summary about that. So what you make on that video still stands today. All this probiotic for this hobby, mostly just land-based bacterial for tooling up whatever. These are garbage cleaners. They're truly probiotics. If you look at the probiotic definition, they should not be considered as probiotics. So in terms of combating coral disease, so for the ocean as well as for this hobby, the ocean is a little bit step ahead because of all these millions of dollars poured into it for the last decade. So for the hobby, we're looking at a blank vacuum zip so far. So that actually can be a very interesting thing. And also with that, many species of coral actually extinct from the wild. And there is like coral bank, like the art of coral is developing throughout the world. And these coral, they will be kept at captive environment. What happened? They get sick, right? What they will get sick is not about when, it's not about if, it's when. So if there is a probiotic developed specially for treating coral in captive environment, then that is a lifesaver for those coral bank.
If we get that, we will let you know. Absolutely. And there's, we're aware that like from this data, we could find something that is that interesting. It's just that's not our initial goal, but yeah, if we can like get to that and we can share it and find some way to sort of just, that would be amazing. And I think this would be a good data set to do that with, potentially. And so I totally see what you're saying, that it would be super valuable. And it's funny, you're talking about the probiotic space, like in the in the human space, it's very similar. Like there are not.
Yep. Yes.
Man, it's like they inherently select bacteria that are easy to grow because it's easy to scale and make a lot of it, and that are pretty consistent in their growth, and exactly, and cheap. And so you end up with those in your probiotics. And so I don't, like I said, we're not in the game to do head-to-head comparisons and any of this stuff. But I see what you're saying. That like I think the next gen probiotics, what I've been saying for a while. First off, they're going to be symbiotics. They're going to be combined pre, pro, post. That's not just going to be a single bacteria. And they're also going to come from this data-driven, like we grew the thing for you. We grew this thing that's going to work in this tank, and here's how we know that it's going to work.
Let me put a comment here from our friend Mr. Chris Meckley from ACI Aquaculture, and he's talking about I've seen this in the chat here, this tulip polyp syndrome. I have no idea what that is. I've never seen it.
Me neither.
But I'm so glad you guys also don't know.
Yeah, but I think the thing that I wanted to point out here in this comment is that there's been a debate within the hobby about lighting and the potential impact that LEDs could have on corals and their health. And some feel that metal halides and T5s, have more beneficial. What's the word I'm looking for?
So
spectrum, right?
Yeah.
That,
The
so-
UV.
Right, the UV, the whole debate about UV and the health of corals. Does that come into play at all in terms of this analysis, in terms of the type of lighting that aquarists have over their tanks and the corals that they're sending into you guys? Are you looking at that as one possible thing to as a variable, the type of light, and potentially that could come into play in terms of LEDs might be not as good as more natural lighting?
Yeah, so we've talked about, or Salem, were you about to say something? Sorry.
Oh, no, yeah, take it, yeah.
Yeah, yes, we've thought about that. It's funny, we've thought about it from like the bacterial angle, because when we grow phototrophs, so photosynthetic like cyanobacteria, which can be not great for corals in some cases, we obviously use like big vats with very specific wavelengths of light irradiating them. And obviously corals also being photosynthetic, you would expect there to be similarly variable effects in growth and sort of health because of the light. I actually don't know if our survey, what it says right now, as in terms of light and what lights you're using, I think that would be an awesome study and idea, potentially hard to do with this cohort, but something that's nice and wonderful about like the fact that we have such an amazingly invested community getting involved with this project is as we are sampling things, if I go into the sequencing data and I'm like, wow, there's this weird photosystem like gene that is showing up in these 10 samples. Why is that happening? I can then reach back out to people and say like, hey, what lights do you use? And my guess is folks would be able to answer that. And that's how we can form hypotheses. That's why I love like microbiome sequencing data, because you can use it to form hypotheses that you can then test in a real experiment. So if we did get the millions of dollars, we get enough data to ask that question, and we go back and say, all right, everybody send us 10 samples from this one species from different kinds of lights, and let's see if that actually, how that changes the microbiome, because that would be amazing.
Mm-hmm. That answers a lot of questions, actually. That they will end a lot of debates.
That would be great. Let's do it.
Yeah, it is.
Yeah.
So, hypothetically, also it's a possibility that if you find all the samples you received from various reef tanks under various conditions, and then you end up having the type of bacteria is pretty common.
Mm-hmm.
So in that case, will that be a logical step to set up a lab that have your own reef tank, so that you are not relying on other people's sample, but of course that you go through the round of other people's sample, right? So in case that you be able to set up the that captive environment in your lab, and that really would be fascinating to accelerate the project a lot, right?
The second one of y'all wants to let me set up a cot in your tank room, I'll go Colin, just say the word. I'll fly down to Miami and be right there. But yeah, you're right. That would be incredible.
Yeah.
That would be absolutely incredible. I would love to, we have lab space, we can do that kind of work at some point. But it's wonderful. So we're based in Colorado. I'm physically in Boston, but all of our microbiology happens in Colorado. So Salem being down the road, feasibly we have the expertise nearby as well.
Yeah.
To be able to show
up
us
a
how
reef
to
tank
make
for him.
That would be awesome. Yeah.
And also that we can donate coral to you.
That would be incredible.
Yeah.
I mean,
the living coral, so you will end up with a collection. Actually, forget about Rebuilder Studio. Right, Salem? You're gonna have your own.
No comments on that there. No comment.
So you basically you're gonna have what Jake Adams used to have or more, and then that will be a community project to support you to have that laboratory.
If someone would like to fund that, I'd be very thankful. I've got a bedroom one right now, so. But there's been talk about with the local North Colorado reef clubs, and there's one of the best shops in the country, Underwater Wonders, is a hop skip away from CSU, about 5 minutes down the road. So we have talked about, like Braden said, when there's extended funding, we'll probably have a big sequence-a-thon. Everyone in Colorado is very excited about this, from all the way down to the Springs and more south up to Fort Collins. And people in Wyoming and whatever else. So if we do get the money, it'll probably be a very large community event, and everyone brings a frag, and we'll have some fun.
Yep.
But it's a money thing.
We
So,
Good to ship
the
you
other
frags.
cool thing,
yeah.
amazing. And the other thing we can do is, like, if we had in-person events like that, you know, it would be so fun to show people how we process corals and how we culture and how we sequence, right? Because like that stuff is portable, and so we can sit there and, you know, okay, maybe it's not the most exciting thing to see in the world, but you can see, like, this is what loading DNA into a flow cell looks like, and here it is actually being read real time, and here I am on the computer watching like the strains of bacteria, so the subtypes of bacteria coming out and saying that one's that one, that one's that one, there's a coral, there's like a fragment of DNA from the coral, all of that. We could do that in a parking lot. It might be nice to, like,
it's real
have a
time
roof,
as well, right?
yeah, exactly. You can do it. You can do it real time. You can. It's so fun. And I mean, it would be fun to see. And also, I'll bet everybody would have advice on how we are processing corals. Because as you guys probably know, so Blake, and this is something Blake and I talked about. Blake, Raquel, Valerie, Paul, a lot of the folks at OSU, we all have different methods in how we prep corals for microbiome analysis. You know, there's like airbrushing to just get the mucus off. There's taking the whole thing and smashing it. That's kind of what we prefer. There's like all these alternate approaches. There's differential centrifugation, where you spin it at different speeds to basically separate out the coral from the bacteria. There's filtering. And it's like it's a whole other study in and of itself. And it would be interesting to see what people say, like, oh, you're doing that wrong for that species of corals. Or why are you doing that to a soft coral? You should just be doing this. That would be incredible.
And to that point, you know, when you're talking about the importance of grinding up the actual corallites, I mean, it seems like most of the attention gets focused on the microbiome of corals that's living in the tissue or in the mucus, but the skeletal structure of the coral itself is certainly a whole nother world of microbiome. And, you know, like I particularly like with LPS, especially wild LPS, where you get like a big colony of Symphilia or something, where, you know, when you get a coral that dies really quickly, typically you get like a bright white skeleton, and you know, and the tissue dies off. But sometimes when a coral will die, it's like, it seems like the tissue, you know, there's Ostrobium or other, there's green bacteria, there's probably red, you know, cyanobacteria. There's all these other different things that, you know, and maybe under normal circumstances can live symbiotically with the coral and it's not a problem. But, you know, given the constraints of being in a home aquarium and various different stresses, that these things actually kind of kill the coral from the inside out as opposed to from the outside in. And I feel like we're just scratching the surface, understanding, you know, all of the various different ways that a coral can get stressed out and get into a disease state.
Yeah, I agree. I mean, with it's usually those big LPS, isn't it? I've seen it on Acropora very rarely, and it's usually aquacultured ones. But I mean, like the only papers that really exist with fungal pathogens and stony corals, it talks about, you know, a dysbiosis in the endolithic layer like that, and they live in kind of like a what's the word? Like they lichenize with the endolithic algae. So then like there's partial bleaching and it'll all bloom together, and it's I mean, yeah, we know nothing about that. There's like 4 papers about it, but we observe it so often. So I think that's a really good point too, is what's that?
And
no one even talks about it.
That makes this really hard is, as a microbiologist, like, so I said how a pound of, sorry, it's so gross, but a pound of human stool is half bacteria, right? So half a pound per pound. In coral, like if I grind up an entire coral frag, maybe 10-15% will be bacterial cells, and it's super, we call it low biomass. It's like mostly coral and host, the Symbiotonaceae, zooxanthellae, and then a little bit of irises, and maybe if there's like a fungal thing going on, that's a whole other can of worms, proverbial worms. And there's a little bit of bacteria, but that's just a tiny bit, and it has this outsized impact. But when you get into the actual skeleton of the coral, I mean, that's really, really low biomass compared to the mucus, which means it's great for culturing, because as I said, like one cell, you can culture it. But for DNA sequencing, it becomes like challenging. I spent like a year and a half of my life developing this like software workflow and our processing workflow. So a big set of algorithms for like pulling out all the eukaryotic and bacterial and viral DNA, separating everything out just so I can get to high quality bacterial genomes and see what's there. But that's one of the complexities of this ecosystem, is that it's really sparse bacterially, but they carry a big weight.
Here's a question, another question from a viewer that I'm not going to begin to attempt to try to pronounce some of those terms there, but Nautilus macro is essentially, wondering, can probiotics be cultured for coral aquaculture?
Great question.
As a possible, way to be, until saltwater probiotics are developed.
Yeah, that is a great question. So please correct me if I'm like missing what you're saying here. But so what you're listing there is Saccharomyces, which is a yeast, right? Like Saccharomyces cerevisiae is what we use to make beer in many cases. Lactobacillus, which is actually a genus, so like a class. You know how like Latin names like Lactobacillus, and there's another name to name the species. Lactobacillus. That's a bacterial genera that is actually often found in yogurt. And Bacillus, so that is another genera that's kind of found all over the place. It's found in soil, it's found in the ocean, it's found like everywhere. And lactose are also found everywhere. All three of these are common probiotic species in humans, and I think maybe in some coral probiotics, not entirely sure.
Every but 2 or 3. This is the status quo.
Okay.
Very much
this is
so.
the status quo. Yeah. So like they totally, they have denitrifying effects. There's like certainly use, but I have not done or seen any specific studies on these. So I see a comment from Dustin, Lactobacillus. Yeah, so that's like the probiotics you see in CVS most of them are going to be lacto-based probiotics. They're going to be like human-associated mostly strains, but they're all lactose. They're like very broadly distributed genera. So yeah, they feasibly could be used, and as Salem said, I think they're kind of the state of the art. But there's a lot else out there, and like in terms of what's naturally found in corals, which may or may not be. Like that could be a good probiotic. It could be not as good. Who knows? I think you're going to see a lot more diversity and a lot of other organisms.
What about?
So there is one report about there's one paper talk about the lactic acid bacteria that actually be able to help coral to overcome some of the heat stress. Yeah, that's something that the lactic acid bacteria from fermentation. Basically, it comes from this guy.
Yeah. That makes sense.
Yeah.
That makes sense. Yeah. Again, like that's how you make your yogurt.
Tolerant.
Yeah. So that could totally make sense. Again, I don't know that study off the top of my head. Also, I see B. subtilis, B. licheniformis, B. pumilus. Those are 3 big ones that you would expect to see in these. In like probably some of the products you're talking about.
Well, it's also maybe important to point out corals eat bacteria too, right? Like it just as a form of food, as a form of available carbon. And I don't know how I don't know to the extent the science fully understands the coral nutrition and what. Whether there may be benefits from eating this stuff, like some aquarists are cultivating yeast and dosing yeast, and maybe that serves as a coral food. I certainly noticed when I was long using the Dr. Tim's the Eco Balance, and then he makes this other vitamin solution, and if you mix them together, you can kind of culture your own. I'd make a 5-gallon carboy. But after a few days, you smell it, and it's like kind of smells like yeast, like it, and then by the end of it, there's like flocculent, and the assumption is, well, I don't know exactly what's in it. I'm sort of also just like any other reefer saying, well, it's marketed as this, it must be good, but do we actually really know what's in it, or are things getting contaminated from the atmosphere in our culture vessels, or is adding it into the reef? Is it the probiotic enhancement? Is it the carbon dosing in the food? Like, I don't know.
Yeah, that reminds me. Well, Salem, remember the Princeton grad,
Yes.
Alan?
Yes.
Yeah.
Yep.
So that is one of the concoction.
That scares me.
Well, this guy is no longer marketed, right?
Huh.
That scares me. Yeah. Culturing
Oh,
anything
yeah.
in the home in uncontrolled and non-sterile conditions, I think, is very dangerous and should probably not be promoted. That's
Yeah,
all I'll say about that.
Yeah.
I think he
I've
stopped selling that. Yeah, that stopped.
So I put up a comment before about clams that I'm just trying to scroll back to find it. Are clams not going to be a part of this study, because it seems like we're running into a lot of challenges keeping clams healthy in this hobby?
Really? Okay, so this is entirely news to me.
And for Braden, specifically, we're talking about tridacnid clams. When people say clams, so these are photosynthetic clams, the giant clams that are the really colorful ones. So it's
the beautiful, colorful ones. Yeah, of course.
Of
course. One of my favorite things to see diving. I was thinking quahogs initially. Being from New England, I grew up clamming, and I was like, oh my god, the quahogs! No, we have to protect them.
Those are very tasty.
They're delicious. Yeah, Clams Casino, it's the best. But okay, that's fascinating. Definitely not in the scope of what we were thinking about initially, but wow, that would be a cool ecosystem to look at, and probably even simpler in some ways than what we're proposing, because we're talking about all these different corals. I would love to do a study, and again, this is just with the ideal world. We're like, okay, take a single or one or two species of these clams. I don't actually know how we'd process them. Maybe it might even just be a swab if they're really mucousy, but like send that to us, and then we'll do, like depending on how diverse it is, like 15 cases, 15 controls, and we'll see what the deal is with an initial pass. That would be fascinating.
Yeah.
So what exactly is the pinch mantle disease for the clams? Is anybody has any idea about that? That is the major thing happening in this hobby, the pinch mantle disease from clams.
And it used not to be a problem, right? I mean, years and years ago, this wasn't an issue, right? I mean, it wasn't.
But right now, it's pretty much an issue that I heard that even the clam farm, the whole raceway is infected, and then lead to a fire cell to the wholesaler. The wholesaler then have a fire cell to the retailer.
It's LEDs. Let's blame the LEDs.
Oh no, you should blame the sun. Well, actually, for matter of fact, in terms of clam, metal halide definitely have the magic for clam. Oh, you're struggling for under LED. Seriously, it's seriously struggling. But the pinch mantle disease is pretty common these days, especially if you're buying a bigger version of it. And then you're very likely that you have a pinch mantle disease. I don't think people have identified that or even have a solution yet. That is the
Rich is also
gigantic.
Rich is also mentioning anemones being an issue.
Yeah,
that's really so. This is actually maybe worth talking about. One of the initial things we thought about in the study that I think actually we might be able to achieve at small scale is like, okay, we have all these diseased corals. Let's emphasize the mysterious diseases or diseased, in ecosystems. Let's emphasize the mysterious ones, and even applying the kind of DNA sequencing we do to aquarium corals and aquarium ecosystems at scale is something that really hasn't been done before. And I think it's worth taking a minute and saying, like, when we say DNA sequencing, my understanding is generally in the coral space and like in most environmental microbiome spaces, people are like doing what's called 16S sequencing. Have you guys bumped into that phrase before? It's like it's super specific, but basically all bacteria have a gene called the 16S gene, right? And 16S sequencing just sequences that one gene. And that is really useful for telling you generally what's there, especially if it's like low abundance. It's kind of like if you had every car on the highway, if you could like weigh it and maybe look at one of the tires, you could generally tell like, okay, there are some big cars, there are some small cars, there's like maybe this one has snow tires, whatever. But it's like you don't know what's in the engine, right? Because you're just looking at one thing. The kind of sequencing we do is what we call metagenomics, or shotgun sequencing, where we basically take fragments of all the DNA in the population, so like corals and algae and bacteria, and we sequence all those little fragments. And then using software, I can put those together into whole genomes. And so basically what that means is like, instead of saying, oh, this is approximately what's there, I can say, okay, this pathogen is this specific bacterial strain that has only been seen in these other environments on the planet, and here's the draft genome. Or this pathogen is this weird fungus, and here's the genome. Well, that's a little bit harder, but like, here's what we think the genome generally looks like. Does that make sense? Is kind of like how just by sequencing this stuff, we can discover pathogens. Sorry if that's too much detail, but I just thought it's important when we're talking about why we don't know what these things are.
Yeah.
So, Andrew Sandler's in the chat. What's up there, Andrew? So he's mentioned a couple of things, some studies that they're working on brown jelly disease, and they're also doing some sequencing. I believe the comment was about regarding clams. Have you guys tapped into the research that those guys have been doing over at Polo Reef? Braden, do you know Andrew? Have you?
Not personally, no. I've seen videos, of course, and like I was listening to some of your stuff. So Andrew's appearance is here just in sort of preparation for this. And so I feel pretty caught up. It's awesome. It's really cool, but I don't think we've ever met. So nice to meet you. Nice to meet you.
Those guys are doing some pretty incredible work in their lab. And so it's some stuff that they're working on that is hopefully going to be beneficial to hobbyists and I'm assuming also to academics and researchers like yourself.
That would be awesome. Yeah, that's really awesome.
And you can sample Andrew's tank.
I would totally, just with his consent. Yeah, that's absolutely. Yeah, let's do it.
So in terms of the sample size, what are you guys looking for? I mean, what is the goal in terms of that that N? I used to be a researcher, so the N is that sample size. What is the goal? How many samples do you guys want to collect for this study?
Initially we said like up to 100 frags, basically. So.
Oh, it sounds like you're gonna get a lot more than that.
Yeah, I know. Yeah. So it's like, thing is, we've been able to we could go a little bit higher than that. Honestly, the limiting factor there is like I said, it's. Sure, there's the funding aspect of it, but we've been really, really fortunate in being able to find partners with good sequencing and like resources and things like that. It's like it takes a lot of effort to process these corals, and so we've had these things where if at the CSU lab, the grad student there, April, who's amazing, if she receives like 50 corals at once, the poor human has to be up for like the entire night processing them. And so we have like a team of undergraduates and some graduate students who can work out, but we're still we're very early in this process, and we plan on sending out kits pretty soon to folks in the next couple of weeks, and then we'll sort of this first phase will wrap up, and then I really do intend to follow through with this in one form or another, because there's so much enthusiasm. It's very clear there's some massive unmet need just in doing the science, and this is very much meant to be our like, here we are, here's what we can do, send us a few samples, let me show you exactly like how good we are at growing things and sequencing things, and then we can all work together and like build something really cool.
Yeah, definitely. Maybe if you go to Andrew's tank, and you will get 300 samples just by that reason.
Sounds amazing. If he wants to help me process, that sounds great.
Is this project, guys, just for hobbyists, or does it also include public aquariums? It
does. We have a few public aquarium partnerships that we've been working on and reached out to, and there's like 5 or 6. We've been getting like the AZA sort of certification, so they're sort of taking us a little more seriously, and that's in the pipe and going well. But yeah, like there are a couple of aquariums. We haven't. I think we've collected maybe 1 or 2 samples from a few, but they're all over the place. So that's been great. having grown up in Boston, I really want to sample the New England aquarium because I worked at a scuba diving shop during my PhD out here, and I knew a few people who dove in the giant tank. And so in the back of my head, I'm like, well, if I can somehow convince them to let us sample, then I'm going to get to dive in the tank, which is a childhood dream.
So I try to sneak in tonight. I'm in Boston.
You're in Boston? Oh, really? Well, I knew Boston.
Exactly.
Where are you in Boston?
Yeah,
I'm sorry, I don't mean to pull.
Yeah.
Oh, you got to visit Dom.
Oh, that'd be awesome. Perfect. That would be amazing.
Give you 100 samples.
That's perfect.
Oh,
Great.
yeah.
We'll go. We'll say it's 8 p.m. right now. We'll see you at 9. That's great.
So I'm
Yeah,
seeing.
I have my diving suits on and the black mask and sneak into
Perfect.
the New England
Yeah, that's
aquarium.
great. Let's go.
Yeah, maybe I can bring back a baby seal.
That's amazing.
So I've seen a couple of comments about Alan Bo's concoction in the chat here, and Braden, I don't know if you're familiar with this concoction. It's a probiotic concoction that's used to feed corals, bacteria that have been stimulated and enhanced rather than feeding corals straight bacteria. So corals, they eat the bacteria, and the concoction is a grouping of bacteria, vitamins, amino acids, enzymes, probiotics, and a carbon source. So there's been a lot of debate about the concoction in the hobby in terms of whether or not it's beneficial or what have you. Is that the type of stuff that's entering into the equation in terms of the work that you guys are doing here in terms of trying to force feed corals certain things? Or are you just more on a mission in terms of researching what is causing the coral disease and try to develop those probiotics to help prevent that?
Yeah, I mean, I think it's, being as you know, having been in research, scientists were incredibly pedantic and really not fun at parties. And so that means that when I think about force feeding corals, I wanna know sort of, okay, what does that specifically mean? Does that mean taking the coral out and dunking it in a tank of stuff? Does it mean, I don't know, it could mean a bunch of things. I think generally speaking, we are really interested in what I would call deploys. Okay, is it going to be a hydrogel? Is it going to be the bag approach? I think Dong, you mentioned that earlier. Is it going to be just sticking a PVC pipe in the tank and pouring probiotic in there? And I have no idea what the best treatment is going to be. My guess is same silver bullet philosophy. It's going to depend on the disease, depend on the coral, depend on the tank, depend on all these different things. If you mean force feeding, just combining a whole bunch of stuff together, or culturing a group of microbes and adding in nutrients and adding in other stuff, if it's science backed and doesn't hurt the ecosystem, that's cool. And by science backed, I mean there's a reason you're doing the thing you're doing. Mm-hmm. Then great. And also and it doesn't hurt the ecosystem is a really important one, obviously, some kind of QC. But I don't know much about the concoction or anything like that.
In terms of the QC of this, it is people using leftover coral foods, dried ones, which have whatever in them, and then other commercial products, and mixing it, microwaving for sterilization, and culturing what they can from that as a kind of on the shelf on a counter, and then pouring that in the tank.
Interesting. Wait, so is it when you say they sterilize the coral stuff and then they inoculate with something else to culture from that?
Sterilize the water and then add all.
Gotcha.
Sorry.
I mean, because to your point, I will say, in terms of growth media that you grow bacteria on, you want your media to match the environment as much as possible. So if you wanted to make a really cool culturing bacteria, grind up a coral, sterilize that, and then you might be able, and maybe add some mucus back in or something, and then you could totally use that on a plate or in a tube to try and grow bacteria that would like to grow on that particular substrate. That makes a lot of sense. I think generally, as Salem was indicating, with anything that is what we would call an undefined consortia, scalable QC is hard, right? You'll know that most probiotics, that even if they have multiple species, what's happening is they're being grown in different tanks and they're being combined together. Because if you take a bunch, imagine take humans, right? Well, the FDA is not going to be thrilled if you said, okay, well, I grew a bunch of stuff. Eat this. Actually, that was there's a really an amazing and sad story about that with fecal microbiome transplants, right? Because they work really well. But for treating C. diff, if you're familiar with that gut infection.
Yep,
but
definitely.
People also died. People died from that. And so the FDA
Mm-hmm.
put a kibosh on it for a little while because it was kind of, it was risky. And so I think there's arguments to be made for enrichments, especially like sterilized postbiotic ones. I like that a lot. But you need to be able to do QC and know what you're putting in there isn't hurting anything. Is that fair?
So, yep, absolutely. Basically, in a very harsh way, is garbage in and garbage out. Yeah. So that's what the concoction is. Yeah.
Gotcha.
Yeah, exactly.
Yeah. But
So
we say that with data too. I mean, that's the important thing about sequencing data,
yeah.
right? It's like if you don't know what's going in, it's hard to get something.
Yeah, you don't know what's going in and you don't know what's going out. So yeah. And also the most important thing is that using land-based bacteria like Salem said. I think I watched that video many times. Wow, that's a great video, by the way, Salem. I watched your video for that one several times. I totally agree. Yeah, if you use a land-based bacteria and grow it out, it probably has nothing to do with this at all. So just like the MCH17 that they grow out the probiotic from the Smithsonian, the first criteria there is a marine-based bacteria as a probiotic for coral. That's first criteria. So the marine base is really, really the key.
Yeah, I'm in the same, like you gotta take something from the environment you want it to work in. I mean, I'm really excited in the human space about human probiotics that come from the gut, and in the animal space and in the coral space and in agriculture too. I mean, we do have a long history of moving things around and sort of trying stuff, and that's fine. But we have the tools now to make it extremely precise,
Mm-hmm.
One of the most interesting things that I've little tidbits of information as it relates to both humans and corals is that there's the disease, white pox disease. All of these are very vague descriptions of disease. It impacts elkhorn corals, the Caribbean elkhorn coral, and the bacteria Serratia marcescens. It was basically they attributed the disease to this bacteria, but it was coming from human sewage that was leaking from the leakage, the sewage systems in the Florida Keys and other Caribbean islands. And it was, I believe, from what I've read, it's the first time that it was documented a human pathogen infecting an invertebrate. So the human microbiome, the coral microbiome. We clearly share certain species, and I think that they're also probably pathogenic for humans too. But to me that's like a really interesting. We tend to think these worlds are completely separate, but actually there's overlap, and that was one of the reasons why the Florida Keys got everybody's on a sewer system now. They don't allow septic tanks because they leak, and obviously there's a lot of other nasty stuff besides the bacteria, but it's an interesting fact.
There's actually that's an interesting idea that, I mean, you see pathogens cross the dome, like jump from branch to branch on the tree of life a lot. And I actually don't know the degree to which we observe that in quote unquote beneficial bacteria, because it's harder to measure. But the Serratia thing is interesting, and obviously in humans you have viruses and bacteria that jump from human to animal to other parts of the world as their ecosystem. And interesting, there are all these fundamentals of microbial pathogenesis, like Koch's postulates, if you've heard of, which is sort of how we evaluate something as a pathogen. It's like, how does it transmit, all this different stuff. Most bacteria that make us sick do so accidentally, in the sense that there are actually pretty few obligate pathogens that want to colonize a human. Like chlamydia is an example. A lot of STIs are examples of bacteria pathogens that evolved to colonize us. And whereas Vibrio cholera makes you sick because it's trying to get out of you. And so I kind of wonder if this is true. And with I'm telling you, microbiome is disgusting. You just got to kind of roll with it though. But it's, and then with corals, I do kind of wonder if we see a similar thing, like these pathogens, they get into a coral and they're not evolved to be there, but through some weird evolutionary thing, they're able to hang out there and use the energy sources of the coral and reef to survive. But then are they just trying to escape, or are there obligate coral patches? I don't know if anyone, I haven't seen much, but who has the time to do evolutionary biology of bacterial pathogenesis in corals, right? That would be, again, if I had infinite money and time, that would be super fun. That would be so cool, right? It'd be important for like the idea of how pathogens evolve. I'm sorry to everyone.
So
in the
next
chat
time
for
that people will say, "Oh, palytoxin is not scary. It's the bacteria from your coral."
That's right.
Wash your hands.
Yeah,
Wash your
exactly.
hands,
Seriously, yeah.
I'm assuming this is not in the scope of the study, but I think I mentioned this before in terms of certain things that could stress corals out, like certain key parameters being out of whack or trace elements being low for this, that, or the other thing. Is that going to be something that you guys might look at down the road in terms of like what would be those key things that hobbyists should be
absolutely.
be aware
Absolutely.
of? Okay.
And we can learn a little bit about what's going on in your tank, I think, based on the bacteria that are there, at least certainly in the water and probably on the coral too, because you can see like, oh, here's a gene for nitrogen uptake, or here's a gene for like respiring iron, like literally breathing iron. Here's a gene for whatever else might be. So that can we'll be able to, it would be kind of cool because I could say, hey, like there's a lot of, genes for using iron to breathe in your tank. What are your iron measurements if you use a little aquarium test strip? Like how high? That would be really, I'd be fascinated to go back to the community and kind of figure that out to a certain degree. Because so that's definitely in scope. A lot of that stuff is in the survey on the site, I'm pretty sure.
It makes me think that, since you're obviously being overwhelmed with people that want to provide samples, that there are already a number of reefers out there who are doing ICP water testing, paying for it. Having it done, if you need to restrict while also getting better data, to take the samples from the people that are doing regular ICP testing just to see if there is any correlation between, trace elements and chemistry and alkalinity, all these different things that have a factor.
That could be awesome. Also, also the other thing that's interesting is in our culturing media, when we grow bacteria and grow these communities out of the corals, we are tailoring that to the environments they came from, right? So I say iron because we do have like iron limited and iron iron depleted, and then with our added media, we have like DMSP to mimic coral mucus. We have this different stuff. And one of the interesting small results that we've gotten just in like the pilot studies we've done, and I wish April were here because she could speak specifically to what we're looking at. It's like, we've seen, and I'm just making this up as an example, but this is the kind of thing we're seeing. Like in, say, the iron-limited coral mucus media, we inoculate a sick coral, whatever that might mean, and a healthy coral, and then we do that in like 10 different media, and you say like, oh, this grew from the healthy coral in this media that has nutrients that coral need, and then in the same media, nothing grew from the sick coral. That could be a really interesting species to try and replenish the microbiome, right, of the sick coral. So it's like if the hypothesis is that, oh, the sick coral is missing good bacteria, which I'm not saying is actually true. Let's say that's the hypothesis. And then I grow a bunch of things from a healthy coral in coral tailored media that don't grow in the sick coral. That would be like potentially a missing bunch of species. So we have some data like that, and we're sequencing it to see what those are. And again, that might end up being a bunch of bacilli and lactose, but I kind of doubt that's going to be the case. And those are ones where I would love to be able to say, all right, well, if anyone has like a coral with the exact same phenotype that seems to be as the one that didn't grow these things, let's see if we could just, use the bag approach and see what happens if we try and restore them. And that's kind of my dream, like rewilding test. Can we re-aquariumizing, I suppose, is the correct way to put that maybe. Because then if
this
that makes
project
sense,
can be so much stuff that can branching out from that. This is
I know, man. Yeah,
so much, so great.
There's so much to do. There's so much to do, and it's like in the data's. I'm really, as you can tell, I'm really excited. Like we have a whole bunch of projects going on, and this is. I'm really pumped about what we're going to be able to do with this one, and no matter what, we're going to find a way to carry it forward. Like we have a whole coral initiative, we have we have the aquarium aspect, we have the ocean corals, and we're gonna figure out a way to let this live beyond just our 100 samples or whatever it is.
So Frugal Reefer John wants to know the panel's thoughts on the purple non-sulfur bacteria that's being used in the hobby, and it's what sold by Ken at Hydrospace. Any thoughts on that?
I don't know, Salem, do you have thoughts on that one?
Yeah, I think it's probably probably the closest tool we have to something with disease prevention or treatment right now. And I say that because Rhodopseudomonas palustris is part of the same taxa, which is often an opportunistic pathogen with many coral diseases. So I can see it as a feasible mechanism that if this does colonize a tank, there could be some kind of competitive exclusion of those pathogenic groups. Similar functions there. I think the more interesting product is Ken's Deep Cycle, which to my knowledge is the only actual reef endemic isolates on the market. So he went to French Polynesia, dove on a reef right by some Porites, and took samples and cultured from that. And it's a mix of green sulfur, purple sulfur, and purple non-sulfur. And there's a lot of evidence that those three core microbes that make up that consortia can do a lot for coral, and some of them are even they're even vertically transferred when coral spawn, at least some species. So I think that Ken is doing very good work, and I think that he is the closest to this world of tomorrow of more targeted therapy with probiotics. Better
So
than
the key point is that
approach.
these are the marine origin bacteria.
Deep Cycle is, yes. Yeah,
Exactly,
Hydrospace Deep Cycle, his other product that's not PNS ProBio, is from a reef in French Polynesia.
Okay.
And it was a reef that was slightly eutrophic, so that he did tests on the nitrates, phosphates, were on the collection point, and it's really similar to what we keep things in our environment. So it's probably, you know, the closest, it's better than a really oligotrophic, nutrient-poor reef, honestly. So I think that is, if I were to have a best or recommended bacterial product in the market, I think that's the one.
Terra Reef Aquaculture just wants to kind of get some clarity, I guess, in terms of whether or not this data will be
that's a great
anonymized for
question.
sure. Yeah.
Yes, yes. To be clear, yes, it will be anonymized. Within the study, if you go to like our sign up page, you can see who signed up, but not who contributed, because again, enough people have signed up that we can't request corals from everyone. So the data is anonymous. We are not like, except for Keith's aquarium, obviously, we're not going to disclose where anything came from. No, we're not going to disclose where anyone's aquarium came from, where anyone's sample came from. We're even like down to, I mean, yeah. And if let's say hypothetically we found a result where, and this would be shocking, like there's a Boston coral microbiome. We're like, oh my God, every frag across every species we got from Boston has the same core microbiome.
It came from Dong, so you know. I mean,
that's right. Yeah, it's all this one tank. If it's one tank, then I'll just tell Dong and be like, "Hey, Dong,
I've got it.
I've the same thing."
I've got Dong's corals. I gave them back to him, and he's given them to this.
Oh, that would be so cool. Imagine if you could do like the lineage tracing. That would be wild. Well, let's say we had that information. We would go back to the specific person who gave us the coral and say, like, "Hey, you know, we discovered that there is like a secret geospatial coral variation. Like, what are you comfortable with in terms of disclosing where you are?" Because we would say like the state or the city or something like that. But that is not gonna happen by default. The plan is literally just be like, tank one had this species, had this microbiome, had these conditions. There's no need at all. I think Terra Reef who asked that, like, we're in all seriousness, we're not going to disclose what's in your tank. outside of to you, of course.
Dong, you had a few questions, and a couple of them, I'm not sure. I don't think you've asked them, but there's one you had in particular about whether probiotics could eventually replace antibiotics, and I thought that was a
great question.
Good thing to talk about.
Yeah. So
Awesome.
one example is right now that in Florida they used to use the amoxicillin to treat the white band disease, and then that price tag is what the hell? If you look at the disclosure, public disclosure, and I want to go make my amoxicillin, God, just so expensive to do. So that is why one of these probiotics goal is to make it really inexpensive. So eventually, if that the probiotic, if that can be totally eliminate all these amoxicillin or Cipro, whatever those kind of freaking
Yeah. Yes, we hopefully I would love to move beyond dosing antibiotics in general, the degree to which we do. We are, as you all probably know, antibiotic, like literally anti-life as opposed to probiotic, like, you know, the opposite.
Pro-life.
And so that, yeah, I guess, yeah, I was going to say, well, that's not what I meant, but it's, you know what I mean. But the point is, it's like, so antibiotics like kill bacteria, right? Or they stop them from growing. And if you use too many antibiotics, you get antibiotic resistance, which means the bacteria will evolve literally so the antibiotic doesn't do anything anymore. And we've seen that happen with penicillin. So like if you get treated with penicillin, you need to, it might not work for your particular like strep throat. And if you're dumping amoxicillin into the ocean, you're definitely going to see that as well. So I do think you can have probiotics or prebiotics or postbiotics that move a community towards a sort of a less pathogenic state overall. And that is absolutely something worth striving for. Or the other option is like if you have bacteria that make really specific antibiotics that are specific to the pathogen in your tank, then you can, and you're not dumping them into the tank, you're like using anything, the bag approach, a gel, whatever, to put it on the disease, then like that is less risky, right? It's about smarter deployment of those systems. But yeah, amoxicillin is, that's why. The other cool thing is phage therapy. Like there's a whole other world where it's like you can use bacteriophage, a whole other thing, bacteriophage viruses that attack bacteria, right? And so they
I think
are
Andrew is working on some of that, right? Andrew Sandler? Is it? Because
Sandler?
Andrew was talking about bacteriophage.
So, yeah. Yes, I
Yeah,
think they are.
so that's being proposed as another method. I don't know about it in the coral space at all, but as another method to attack to like in human to replace antibiotics, right? Because bacteriophage live inside the bacterial cell, and when they replicate, they lyse the cell and kill it. So you can target that virus to that one strain of bacteria, and it will theoretically eliminate it. In theory, reducing the risk of resistance, depending on like the mechanism through which it's killing all this technical nonsense. And then it also doesn't have this broad spectrum effect where amoxicillin like kills half of the bacterial tree of life or whatever it is, because it attacks a conserved part of the bacterial cell. So that's a whole other thing that's exciting.
Yep.
Getting
And
back
you can make a human antibiotic out of that as well. A new generation.
Yeah, if you find new stuff, I mean, it's we need more. We need more human antibiotics. We have not found many new ones. Yeah.
Don, you had another question about using probiotics to potentially help prevent, helping corals with high temperatures.
Yeah, the heat stress.
The heat stress. Yeah, is that something that is also within the scope there, Braden?
Yeah. So for that is Herkel Peixoto, who's a PI, a professor over at KAUST, was and they built the oh my gosh, I've literally dove there. It's the Coral Reef Garden, the Probiotic Village. Probiotic Village. Sorry, thank you. Oh, that's embarrassing. The Probiotic Village. Herkel she's one on our scientific advisory board. Wonderful person, and really, really great professor and researcher out there. So it was like a 2016 Science Advances paper or something like that, or maybe 2019, that had the cocktail that confers tolerance to heat-related bleaching in an aquarium setting. I think that's a really exciting mechanism, and she and her team are definitely the people to speak to it specifically, much more so than me. I don't fully understand the mechanism. I don't know. They have a bunch of actually really creative ideas for deploy, everything from like feeding corals rotifers, which have the bacteria in them, which is a fun one, to like ROVs, to all these different approaches. But that is that I think, you'll hear, I just say the same thing like over and over again. My guess is there is always more to find. There's more strains to find. There's more that are going to work better on different species. There's more deploys. There's a better understanding of like what actually enables colonization. That's something we haven't talked about yet. In the human probiotic space, the state of the art is like you have to keep taking probiotics. And that's also, kind of like what a probiotic company would like. That makes sense. They want to sell probiotics, but the data does show that like probiotics don't colonize your gut. They eventually leave, as you can imagine. And in corals, I don't think there's a lot of really solid data that I've seen where you do these studies of like, okay, how are you getting the probiotic on the coral? How long does it stay? Salem said there's a couple where you look at transmission, right, between generations. That's great. But for actual probiotic depletes, that's a whole other thing.
Yeah.
I just had something that I wanted to ask, and we've talked about pathogenic bacteria, but can the use of probiotics backfire in the sense that it gives pathogenic bacteria a mechanism to multiply? I mean, is that something that's possible? Playing with Mother Nature that way, is that even a feasible scenario, or is that?
I think it's more likely with antibiotics, because probiotics you're adding something, and there's always a world where you could disrupt the natural ecology of an ecosystem and have something bad take over. That is with antibiotics that happens. Again, I'm going to keep using this example of like C. diff infections in the gut. You take antibiotics, you have one cell, right, of Clostridium difficile. It can take over and become your gut microbiome, make you really sick. And that could totally happen in corals if you throw amoxicillin onto the coral. It's going to kill everything. And then if there are pathogens there, and your environment and your tank shifts to one that is more selective for, as Salem said earlier, opportunistic pathogens that take advantage of the opportunity to be a pathogen and make your coral sick, they might bloom. With probiotics, I think it's a greater risk actually if to sort of return to what you were saying before, if you're not entirely sure what you're putting into your sample, into your tank. Because if there is pathogen contamination, or if there is something that can have a deleterious effect, but no one's like done the study on it, then that could be really problematic. I think the idea that you could have a network effect from like a bunch of probiotics that change the community in a way that could happen, but oh my gosh, we don't have that's some hard science right there. No one's ever cracked that for like any complex ecosystem before, and because it happens so infrequently where you'd have like probiotic leads to pathogenic effect.
So since you mentioned seeds, sorry about that. Sorry, go ahead.
I'm just going to throw in a quick question. You go ahead, Don. You finish your thought. Go ahead. Finish your thought.
Since Braden talked about the C. diff several times, so for people who are interested in that, you can Google it and watch the video of what those kind of thing, especially the horse. It was. then you will see that what kind of disease that is. So basically you just have a diarrhea.
Which is like that's what it is. Same thing with Vibrio infections. It's a lot of, unfortunately, there are not that many bad bacteria. I really want to emphasize most bacteria are good or not really doing anything that is our business anyway. Most of them are good. So there are very few bad bacteria, but they do have a dramatic effect, and you notice it.
Yeah.
I don't know if anybody has an answer to this question from Spin Sanity, but has there been any more conclusive evidence that brown jelly disease is caused by Archibacter and that Cipro can treat it? This was a hypothesis from Aquabiomics around 2020. Any thoughts on that, or?
I don't know off the top of my head. I don't know, Salem or
Yeah, I will say sorry, Eli. I do not buy it. So basically, I think that we probably need to leave behind the idea of a one pathogen, one disease model when it comes to coral.
Yes.
And I think that, like
100%.
Braden has said, the best model that we can think of is one like C. diff. That is most likely what's occurring, to where we have to think of this from an ecological standpoint, because that's what it is. So there is a disruption in that ecosystem, and something takes advantage of that. And some things in the environment can cause a disruption. One of the big ones I've been screaming about is high levels of dissolved organic carbon, labile dissolved organic carbon. That's just one example. Also, low levels of oxygen can shift the viral communities. There's a lot of these different chemical parameters that will then change that ecosystem. And a lot of the good guys really don't like that instability, and guys that are really tough come in and take advantage of that. So brown jelly disease, the Arcobacter has been seen with 16S data in many cases of it. But if anything, I would say it could potentially act as something like a keystone pathogen. If that's the only potential argument I could see for that, to where it comes in and induces some kind of bad gene expression in some other people and gets them all. It's like someone coming in and organizing an army and they all get kind of mad, and then it causes the coral to go down. And this is a similar model to what's being explored with either white band disease or stony coral tissue loss disease. I can't remember which one in the Caribbean right now. But that's it's still a consortium. It's still a pathogenic microbiome that emerges. And I think that that's what we're dealing with here, is instability leads to that. And it's also, it's not going to be one size fits all, because I've seen some data with brown jelly and things that do not have Arcobacter. Obviously, there's many bad players, Vibrios, Rhodobacteriaceae, Flavobacter, etc. But they all have the one thing in common, which is they tend to be copiotrophic, and they tend to have the ability to have pathogenic gene expression, which can be turned on and off based on the environment. So that's the real thing, is there's no bad bacteria until there are. Dr. Jekyll and Mr. Hyde.
Oh, well said. That's a good one. I'm totally going to borrow that. Yeah. But so Salem, that was truly beautifully put. I think, to bring it back to a point we were talking about before, one of the reasons why I'm comfortable going broad here, especially if we have a disease emphasis, is because we are able, with the kind of sequencing methods we're developing and using, to look at specific pathogenic genes versus the species they're in. We can form this hypothesis of like, hey, you sent us, 10 of you sent brown jelly. We found it all had the same like weird type 6 secretion system, but it's found in 5 different species or strains. That'll be the kind of thing we can get at. Anyway, that's Salem. That's a very, very good point. And I like the Jekyll Hyde thing. That's spectacular.
But yeah, it's about gene expression and what genes are expressed, and many of those things can be conserved, which also gets to how does this transfer over to the wild. Likely those same genes that are virulent, that are being expressed, is how coral disease happens in the wild too. So in my mind, that's really the holy grail chunk of data that I'm really excited about with this project, is maybe we figure out that those types of mechanisms, and that gives us a much more universal look at coral disease than really any study has looked at thus far, even in the literature.
I mean, has anybody ever documented brown jelly disease in the wild?
I think my feeling is that what is it? It was what Jamie Craig's White syndrome. Is it White syndrome? I can't remember. There's one that also has philaster and all of the opportunistic ciliates that Michael Sweet and Jamie Craig's looked at a long time ago. And that seems to be the kind of closest analog to brown jelly in the wild, is you have all this pathobiont, then the ciliates come in as a secondary, and it's a really similar type of thing. And I think their hypothesis was the flow is high enough, it just pushes the tissue away.
I mean, my hypothesis, because I used to collect Recordia, professionally back in the day, and I'm fortunate that brown jelly hasn't really been as much of an issue. I think because I'm using higher water flow. I mean, I think that's the number one way to prevent brown jelly is having high water flow. Although about once a year I'll come in the torches that were fine yesterday, there's 3 heads that are completely enveloped in brown jelly. But when I would look at them, under a microscope or up close, it was like you see the ciliates living around the brown jelly, and I just sort of always thought that maybe when the ciliates poop, like they are also creating a nasty environment that sort of is just like this downward spiral of which obviously prevents oxygen from getting to the tissue, and it seemed like, to me, it was sort of more a secondary infection as opposed to the primary, infection, and the ciliates are what we observe after the fact. But that was just sort of my hunch, but I don't know too much about it.
That's my understanding of it too, and that's also Michael Sweet did some pretty good papers where they would selectively wipe out the protozoan population with metronidazole or whatever, and then the disease would still persist, albeit at a slower rate. So that seemed to support their secondary. There's some studies from a group in Taiwan, though, that shows direct inoculation of philaster, and that can cause death. So it's really a chicken or egg thing of, who knows what would happen in the wild if you can get that dosage size to have the ciliates alone cause it, which, this will be interesting too, using the shotgun approach, as we can see the protozoan community in a lot of these things. So we could then tease out what's a healthy versus an unhealthy protozoan community. I think that's also, who knows at this point? There's like 3 studies on it.
You can see why I'm so thrilled to be working with Salem and Colin on this, because it would not even remotely be possible. So I wouldn't know any of you guys, and the depth of knowledge both of them have. It's extraordinary. You guys are the best. There was a question about from Terra Reef again about this. I think important. It's what data are you hoping to receive in terms of the typical reef aquarium systems? That's a great question, and it actually comes back to what Salem was just saying. I think outliers, as you're saying, are actually. I think everything's going to be an outlier, and in the sense that that we in the microbiome field we like to say there's an Anna Karenina effect, which is a very pretentious thing. But the first line of that novel is all unhappy families are unhappy in their own way, and all happy families are happy in the same way. We actually think of it as a reverse Anna Karenina effect, where you have more conservation and unhealthy communities in the microbiome. And healthy communities can look like all kinds of different stuff, and they're all going to be their own kind of outlier. However, the exception to that is when we look at the genes that are present. So when we use shotgun sequencing to say, well, we don't know, maybe all of these healthy communities from a species standpoint, they look very different, but they have the same 30 microbial genes. And maybe the same will be true for the unhealthy communities. It'll be even narrower. And so that is where, again, going broad is kind of going to be, I think, helpful for us. If that helps sort of address the question, kind of.
So last question will come from Todd at Champion Lighting, and it's back to the sample size. Is the 100 test sample, that's the question. Is that going to be enough to give you guys actionable data? And
that's a great point. Sorry, Keith. I didn't mean to.
Yeah, no. I guess the question on top of this one is it just budget? Is that the reason why?
It's the big thing. Okay. It's partially personnel, but at the end of the day, we can all sit there and work on corals, and that's part of the job, and we all love it. It's like I said, that budget of we're doing $1,000 a sample with sequencing and everything like that, all in, and we can get it down through a bunch of sort of clever things. It gets expensive, right? And so it's 100 is enough to form preliminary data to raise more money through some mechanism. It could be, there's a whole bunch of different ways we could do it, but I feel pretty confident, especially just from the enthusiasm of the community, that there's an unmet need, and therefore with the data we're going to get, we can be really well positioned to help carry that forward. But that's why, and it's personnel and funding. It's, this is a pilot project, we would consider it. And then as we move into 2026, Salem, Colin, and I, and the rest of the team will put our heads together and be like, all right, how are we going to get 1,000, 10,000, 100,000 samples?
How many samples do you guys have right now? In terms of willing participants, let's put it that way.
I think in terms of participants, there's 50 some people signed up on the site. A bunch of y'all, I think. I don't know, Salem, if you mentioned to somebody, something happened in the past 48 hours, and we had this spike of signups, which has been amazing. And then we'll keep. There's also a great documentation side of this, because it's really amazing seeing the photos of everyone's tanks and kind of getting a sense for even something that we can do where we go above 100 samples is if people are willing to fill out the survey and just say what they're doing. Being able to know, this is the range that there is, or this is the range of what people are seeing. Take, Colin, you were referencing, or you were talking about brown jelly earlier. How many people have brown jelly showing up in their tanks? What's the range? Things like that. Now, of course, again, when we publish any of that, that's all anonymized. But that kind of information that's just quantitative and qualitative, but doesn't require DNA sequencing and analysis, can be super helpful. And that's all. You can go join that on the site.
What's the website people can go to sign up?
So you can go, it's twofrontiers.org, and it's all linked right there, Project ReefLink. But I can send don't know. Okay, am I able to access the chat? Here's
I sent it in the private chat. It won't let me post the link.
Oh, okay.
Cool.
Let me put it.
Okay.
Let me put it in the comments right now.
And if you have questions, please reach out through the website. There's a little comment forum that I literally get a notification on my phone, or you can reach out to me directly. It's my first name at twofrontiers.org.
Let me put this in the chat.
Braden,
Sign up here.
You know, I've sent a few different batches of samples to kind of get the whole thing going, and one of the corals that I sent was a sample from a Bacillopora damercornis green, nothing super special, but we're calling it the immortal strain because one, I have not experienced any RTN on this one genotype, and you know, although I did years ago, I had experienced it, but I haven't experienced it ever since, at least since I sent the sample. But I'm interested to kind of you know, see where that leads. Obviously, there's no such thing as a bulletproof coral.
Yeah, we have data on that. I actually think we have. I think we have sequencing on it, but we have culturing from that as well. And we've talked a little about some of the interesting isolates that we will in turn be sequencing. So yeah, we've. It's amazing how much you can get just out of a single coral. There's a lot there, and yeah, that's a really cool one.
And so run people through kind of like what was like. What are you doing to then, you know, then find, hey, you know, this coral had some isolates on it that proved to be resistant to, you know, certain pathogens, or what is it that you do?
Great. That's a great question. So the first thing we're doing is going and sorry, I'm distracted by the chat. But it's basically we take the sample, we process it. As I said, we split it for DNA sequencing and culturing. The first most obvious thing we can do is say use our, like, we have a bunch of custom algorithms and our custom sequencing approach is all very customized. And say here are the strains that are there, and that is very fast turnaround. The second it comes off the sequencer, we can basically have it done, and then that is the first thing to say, like, hey, this coral is different. Or if we identify those core genes like that, being able to say these are the 30 core healthy coral genes versus these are what these other ones mean, those biomarkers, basically, we'll be able to, even with 100 samples, have that kind of panel and data put together. Then the next thing is for culturing. As I said, we put into enrichment culture where we grow communities of organisms, usually a couple of species in these different specialized media to grow things that we think are going to be relevant to the coral ecosystem, generally speaking. We preserve and we sequence those as well. And then if we see something really interesting, like say a lot of growth from your immortal coral, but not from one that is really sick, we pull out single isolates and sequence those as well. And so what we can give back at the end of this whole process is like, here's what your initial sample looked like. Here's what we grew from it in mixed culture. Here's what we grew from it in pure culture. Here's ideally how it maps back to our reference database. And then, you know, the cool thing is we have everything cryopreserved. So let's say in the sequencing data for the healthy coral, I'm able to say, oh my God, there's like the magical coral strain in there. Or maybe there's like a version of that ocean strain that they're using for white band disease. We can go back and design conditions to grow that exact organism because it's all sitting in the freezers. So we can then try and grow it out and do the same thing. And that's how the really value, and I can't believe I didn't say this, the value like tool, the valuable thing that we do here is like we preserve everything for culturing in perpetuity, again, scientific resource, open to the scientific community, and then also we sequence it as well. And those two things inform each other and ideally inform you. So what I can say is everyone who sends us a sample, we are going to do our best to sequence it and, you know, ideally culture from it as well. Now that can fail, right? Obviously, like things can always happen here, but we'll sequence it, culture from it, and tell you what we get because and see what we find.
Well, guys, Braden, Colin, Salem, thank you guys so much for this really very educational, enlightening chat. So much information. And really appreciate the work that you guys are doing to try to solve this. And when do you guys expect some actionable results to be available to the community?
aiming for the first couple months of next. Well, let's be generous about it. Let's say like Q2 of next year. We'll certainly have some data back in terms of some sequencing and stuff like that.
Cool.
We're still getting samples in, but we're called the second a sample comes in, we start culturing. So if you really want to know, like we can give you some feedback as soon as stuff starts happening.
Awesome. All right. Well, listen, thanks, guys. I also want to thank our sponsors one more time. Polo Reef. Make sure to check out Polo Reef's latest YouTube videos. The Polo team takes on another crazy month after ICP test results reveal toxic metals. The team continues to solve the issue. The team fixes the mess from the rock slide in the 17,000-gallon tank last month. Oh, boy. And all the while tackling lighting issues, saving rare fish, and finally fragging giant acro colonies, exposing the dirty side of reef keeping. No one at Polo Reef was happy with the smell. Make sure to subscribe to their YouTube channel at Polo Reef for video drop notifications. Champion Lighting and Supply. Todd, thank you, man, for the little gift here in terms of the Reef Delight lens port and coral lenses. I'll play this video one more time. I shot it earlier today on my phone. So this is a great way to take incredible underwater shots using your smartphone. So it's if you want to check it out, get more information, visit championlighting.com. I want to give a big thank you to Paul, who's also the moderator, as well as the president of the Boston Reefer Society. Please join and support your local reefing clubs. They're so important to this hobby. Also want to let you know that all episodes of Rappin With ReefBum are available as podcasts on Spotify, Apple Podcasts, Google Podcasts, Stitcher, and Amazon. The next Rappin With ReefBum live stream will be on Tuesday, December 16th. Typically, we're the third Tuesday of every month at 7 p.m. Eastern Standard Time. We're going to be talking about lighting. We got Tulio coming on from Reef Brite. We got Luca from Quanta Lights, and we got Logan from Reef Breeders. They're going to be our panel next month, so that'll be really cool. So please tune in for that deep dive on lighting. Until next time, be safe and be well. Later.
Thanks a lot.